thanks very much for the opportunity to speak today. My name is Michael Craig, and I'm a senior scientist at Valence Labs in Montreal. We're actually working directly with, or required by Recursion Pharmaceuticals last year, so it's great to have the opportunity to test a lot of our work directly in real like wet lab drug discovery settings as well. And we've also recently opened an office in London, so if you're interested in learning more about opportunities with us or just want to chat about any of our work, we're happy to speak after the talk, or you can contact us by email as well. And you can talk to Terence Bois, our co-founder, is also here today. But today I want to talk about language agents for autonomous workflow orchestration. So this is one area of interest for us now, is trying to use large language modeling applications, not just treating them as like databases where you can extract information from, but really as orchestrating tools that allow you to kind of think through complex processes and autonomously run workflows in a drug discovery setting. So I'm sure, as everyone here is aware, the drug discovery process is very long, very expensive, and the search space is absolutely massive. So it takes 10 plus years to get a drug to market, and even longer to get it reimbursed oftentimes. Around $2 billion as well, plus this 10 to the 60 number that we're all aware of as well, drug-like chemical space. So this is obviously a massive problem, and all these different steps, of course, can be broken down much further into sub-steps as well. So there's so many different opportunities for machine learning to go in and help improve processes. And oftentimes what you see is people develop point solutions for one specific aspect of the drug discovery process. So part of that can be represented by improving how we represent biomolecules computationally. So this could be small molecules, larger molecules, proteins. Improving how the system can represent those entities is really important. Also, structure-based methods like AlphaFold and diffdoc are also making an impact now. So that they can help on looking at efficacy or binding affinity for a compound with a particular protein. And if you've worked with medicinal chemists directly, they will always ask you questions about how synthesizable your molecule is as well. Oftentimes, what you see from generative models in this space are compounds that can be very interesting and novel, but if the representation is not capturing the stereochemistry perfectly, say, it's difficult to differentiate between one compound that is synthesizable and another compound that's not. And of course, explainability. So if you're working directly with biologists and medicinal chemists, they would like to know why it is your model thinks what it thinks. So there's all these different point solutions for all these different problems in drug discovery. And the question really is, why aren't we seeing this huge impact of machine learning applied to drug discovery? Why is that the case? And there's all sorts of reasons for this, of course. Merging these two fields together is a huge process. And oftentimes from the machine learning side, we see 1% improvements on state-of-the-art models. For a medicinal chemist, that might not interest them too directly oftentimes. I think this is changing as well, especially with talks I've seen today. It seems like the documentation and work on that is really improving significantly. But oftentimes it's quite difficult to actually implement a point solution into a real live discovery program because the scientists working on that might not have the coding background necessary to do this. And from the machine learning side, we're often talking about just generate more samples for them, right? That's often not a trivial process in live discovery projects. There's all sorts of angles for why you make a certain decision of why you're going to synthesize this compound or that compound. From the life sciences side, they're very used to these expensive software packages that they've used for 30 plus years. Oftentimes, this is a linear regression or random forest model trained on a very small set of compounds. And if we've talked a lot today about out of distribution generalization, this is for sure an issue for them when applying these models internally. Again, the synthesizability issue, of course, always coming up. Why can't I synthesize this compound if it's so interesting to you? And really their question is, when is the exploitation going to start? When are we actually going to see real gains in speed and in making the process more efficient and cheaper? And really just to illustrate this further, it's because there are so many different aspects to the drug discovery process. Some of them are not just scientific, but all the different financial constraints that companies have as well. So from our perspective, what we want to do is figure out how we can take all these point solutions and start stacking them on top of each other and figure out how you can go from a drug discovery process to more of a drug design process. And the way we're going about that now is at Recursion Valence is to design a LLM orchestrated workflow engine. So what this system is able to do is you're able to query a large set of different computational tools directly using natural language. So instead of a medicinal chemist needing to learn Python and understand how to use a Jupyter notebook and download a repo, what they can do is just ask a question directly to the system. The system will figure out which tools they want to use, which visualizations are relevant for their different tools. And then even in our case at Recursion, we're able to order compounds directly from this interface and get experiments scheduled on the internal Recursion experimental systems as well. So we're really trying to think end-to-end, how do you go from indication, target identification, all the way through experimenting directly with those compounds and trying to do that via natural language specifically. A little more detail on how we're thinking about this. So LOW is really an early version of a cognitive architecture for drug discovery. And using this term cognitive architecture, I mean that the agent, the central orchestrating agent, has both context for the task it's trying to accomplish, and also the ability to reason about different steps and tools that it can use to implement those processes. You can see on the right side of the screen there's a memory storage and then a wide variety of different services and actions that LOW can call from. And really the long-term thinking for this project too is at first we want to have our scientists both within Recursion and our partnered companies as well to be able to use this system, but eventually we want to figure out how we can make it so that these systems can work in more and more autonomous fashions so that the touch points become fewer and farther in between and that they can directly generate complex reports and serve these to the scientists and figure out how you can drive programs that way. A little more detail, too, on how this actually works is this is obviously still somewhat high level, but really we want to the central agent orchestrator is made up of a central large language model. Oftentimes, this is one of the very large commercial models. They do very good. They're very efficient at orchestrating processes. Plus a set of prompt frameworks which define different decision modules and decision frameworks that the model can use to put together these workflows, and then also access to the descriptions and APIs of all of these tools as well. So not only can we figure out which tools to use, but we can understand how to formulate the arguments for, say, a Python function or something like that. You can format them as JSON strings, pass them to the tool, and the tool can use them for whatever they need to do. In this case, you can ask questions like, design a new compound with the same scaffold as gefetinib and ifatinib, but with better CNS penetration. And the way the system will think about this, it'll start by saying, okay, I need to get those structures first of those two relevant compounds. I need to identify the central scaffold that's overlapping between these two compounds, then figure out which generative model I should use in order to execute this expansion around the initial starting scaffold, and then figure out which predictive models are relevant for the task that they're working on. So in this case, it's CNS penetration. There's various multi-parameter optimization methods you can use to improve CNS penetration. And then as the system is thinking through all these different steps, executing these steps, it's looking for that terminal state. So at first it defines, here's the goal of the user, and as soon as I hit that goal, that hits that terminal state, and then it'll give you the output of, hopefully, an optimized compound in this case. Just some examples, too, of what this actually looks like. So you can start with a kind of a simple example of like, what are the top 10 known drugs for EGFR, say? In this case, we're comparing it directly to ChatGPT to kind of give you an idea of how our system is unique. For ChatGPT, of course, doesn't have access to this tooling. And it does a decent job of identifying these compounds, but it just gives these kind of one-sentence descriptions of what they are. With LOW, you're able to access live databases that are constantly being updated with both internal and public information. And it's able to pull directly from these databases and give you structured output like this. That's much more information-rich. Another example here is if you were to do chemical neighborhood exploration around osimertinib in this case, ChatGPT does not really understand what you're looking for with this kind of question. But of course, with LOW, you're able to access our own internal foundation models for generative chemistry. And you're able to do these different representations of the compounds, look at these different visualizations as well, and you can extract all this information as tables or as visualizations and anything like that. Even when you trick ChatGPT as well, when you kind of try to prompt it a bit more and specifically say, I want you to give me compounds at the end of this, actual smile strings, it does an interesting job of calling the data analysis tool. So it wrote this Python function with RDKit to execute this. But the key issue here is that the compound or the smile string that it's using is not osimertinib. So that's not a good approach if you're trying to optimize osimertinib directly. It's able to do some interesting coding, of course, and it's obviously a very powerful system. But the biochemical entity recognition is not sufficient in this case. And that's kind of highlighted here even further with a very direct a question, no, please give me the smile strings. And it gives you some smile strings, but they are not relevant to the question that you ask. So, and oftentimes the smile strings it gives they look like real compounds, but when you actually try to convert them to a molecule with RDKit, it's not a real smile string in that case. And even further, I can directly give it the smile string of osimertinib that I wanted to use, and it's still a bit confused about what exactly I'm looking for. Where again, as with Lowy, you're able to call the relevant tools, ensure that the smile string is represented correctly across all of these different tasks, and to improve the whole system directly. And I thought it'd be useful to go through a more explicit kind of multi-step process as well. So in this case, what we're doing is we're starting with indication, and we want to get to chemical series. And we're going to do that in about 2.5 minutes. So essentially what we're able to do is start with a question of, are there any, what are the relevant targets for, in this case, non-small cell lung cancer? And these are from the literature. So these are all targets that are well known. There's a lot of drugs against them already. You're able to do these kinds of visualizations, and all this information is contained within the system, so you can look directly at all these different parameters of interest here. From here, what we're asking the system to do then is use Recursion's internal mapping data to take those input compounds and find novel compounds that the Recursion mapping tools are able to extract directly from the map. So in this case, we're identifying a novel, somewhat novel gene, RAF1, which has correlations with several of the input compounds as well. So the system's able to recognize, RAF1 seems to be the most interesting target here. And now we're using a system called Matchmaker, which is another internal Recursion tool, which takes the entire proteome and all of the 36 billion compounds from enamine and identifies those that have that are predicted to have a relationship between the binding pockets on both allosteric and orthosteric binding pockets as well on these proteins and different compounds that are of interest. So from there, we've identified a set of starting point compounds that are publicly available. And what we can do now is ask a question, generate compounds that are in the same chemical neighborhood as this initial smile string. You can watch me slowly type this in. And also calculate their ADMET properties for prioritization. So what we're doing now is calling a generative model, a generative foundation model, to generate compounds that are in the same chemical space, essentially creating a chemical series from that initial starting point compound, and then also eventually generating calling another set of ADMET prediction models, which are all trained with internal Recursion data to identify the ranking of those output compounds. So at first we give an approximation of drug likeness from that. So some of the compounds coming out would be discarded at the first step. And then the second step is putting all of the drug-like compounds into this set of 10 or so machine learning models to give approximations for the different ADMET properties, which help us prioritize this final set. So this gives you an idea of how we can use this to orchestrate processes. I mentioned to increasing autonomy with these systems. Right now, we can do kind of 3 to 5 steps in a row based on the context length that we have access to right now. But I think as that context length increases and the prompt frameworks and decision frameworks improve, you'll be able to really link these steps together, and you could have these things running autonomously a bit more. And the kind of in relation to this, this is how more of a general framework for how we think about building these systems. So you have your user prompt input, then this decision cycle, which gives a proposal at first, evaluates that proposal, selects the different steps to execute in the order in which to execute them, executes those steps, and updates the system prompt so it increases the context of the task there. And you can see the action space as well. So this gives essentially the different options for the model at that first step. This can be software tools, machine learning models, all the robotic systems that we have control over within Recursion as well, and of course external APIs. And what I'm most interested in too right now is these memory modules. So currently these systems don't have long-term memory. But you could think of ways of incorporating both episodic and semantic memory type systems into the prompt framework, so that, say, the model has seen a very similar scaffold last year at some point, or someone else on another team has worked on this particular protein. Well, you can incorporate that long-term memory system and identify the previous work that was done on those systems, incorporate that into the context, and kind of hopefully improve the system overall in that way. So yeah, we talked about language so far, but of course language is not everything, and I think it's fair to say that you cannot use language to represent every single biological or chemical modality. So a lot of what we're doing at other parts of Valence is to build these billion-scale parameter foundation models for several different biological and chemical modalities. Also multimodal modeling as well to improve how these systems think about biochemistry. And all of these will be directly accessible via the low interface as well. And kind of in the short term, how we're thinking about this is, if this is a slightly more complex prompt that we're not quite here yet, but this is the kind of short-term goal with the project is, say you would take a chemical starting point. It's a strong inhibitor of a particular target, and it serves as a treatment to, in this case, it's the EGFR pathway, the resistance pathway in particular. However, it's ineffective in patients with a particular secondary mutation in the binding pocket. Given the binding dynamics observed with this particular foundation model, if you were to make that mutation, can you suggest a set of promising analogs to overcome that issue? So the amount of different kind of biological and chemical representations, plus all the different orchestrating processes, would allow us to kind of ask this kind of question and make progress on this particular drug program. And really long term, it's to push this even further to a point where we not only are able to orchestrate these processes, but also to generate biological hypotheses directly with the system, to then design experiments, run them directly on our own internal platform, learn from those results and update our beliefs and strategies about the system in general and about whatever pipeline project that we're working on. And then, of course, repeating this cycle for 24 hours a day, 7 days a week, hopefully to kind of transition from this discovery process more to a design process and enable large-scale drug development. So yeah, thank you very much for your attention. Happy to answer any questions.